All right. I think we can get started here. Um, cool. So welcome everyone. Thank you so much for joining us in the Polaris webinar today. I'm really excited to introduce Peter, who's doing his second year of PhD right now, studying biochemistry at University of Ljubljana in Slovenia. He's previously worked in Paris at Galapagos, which is a pharma company. And his work is in method development and benchmarking for protein-ligand and complex prediction, which is going to be one of the main topics of today's discussion as well. This is really, really awesome work with the runs and poses benchmark, and we are very excited to host both the benchmark and the dataset on Polaris. I think today is going to be a really awesome discussion just to, have Peter, share a little bit more about the methods and open this up to be a conversation with the community. So thanks so much for spending the time to be here today, Peter, and I'll pass it over to you to kick things off with the presentation.
Yeah, thanks, Johnny, for this nice intro. And yeah, also thank you for giving us the chance to share our work with the broader audience. Yeah, it's really a pleasure. So yeah, the topic for today's webinar is our latest preprint with the title "Have Protein Cofolding Methods Moved Beyond Memorization?" So in this work, we introduced new benchmark dataset called runs and poses with the aim to detect the true breakthrough in the protein-ligand complex prediction. So the preprint and our GitHub repo can be found in the QR codes here, and also the ML-ready Polaris benchmark. So yeah, let's get to it. So this is kind of the outline for today's talk. First, I will start with the introduction and the motivation. So why did we decide that we need yet another benchmark for protein-ligand complex prediction? Then I will go a bit into the methods. How did we define difficulty and success rate for protein-ligand complex prediction, especially for cofolding methods? That's a bit different than for the docking methods. Then, yeah, what did we find out? And also, yeah, a quick discussion, some open questions. If what can be done to change some of the results? And at the end, yeah, we can quickly take a look of how can you actually use this benchmark that we developed. So yeah, first of all, why the protein-ligand complex prediction is important. There are quite numerous reasons. One of them is for understanding the fundamental biology. So to understand how protein function, how they interact with other molecules in the nature. The second one, kind of the most obvious one, is for drug discovery. So the protein ligand prediction can be used in virtual screening, in lead optimization, for improving specificity and also potency of the ligands. And also it can be used in protein design if you want to design a protein that would better bind to your ligand of interest. So similarly as how it is done in the protein complex design. And yeah, currently, I mean, mostly the protein ligand complex prediction is done by the physics-based methods with their pitfalls, of course. What people usually use is the docking tools, where you try to find a ligand pose based on your input protein structure that would have the best potency to this protein. So in recent years, deep learning had changed biology quite a lot. So namely, it had a huge impact in the protein structure prediction and also protein design. So I guess the audience here knows quite a lot about AlphaFold 2, so I don't need to go into more details there. But it's worth mentioning that Demi and John got a well-deserved Nobel Prize for their breakthrough in protein structure prediction with AlphaFold 2. What is kind of more important for this talk is how did we actually know that AlphaFold 2 was a breakthrough? So it was basically because of the benchmarking efforts that has been done over the years, even before AlphaFold 2 came. So there is this conference, Critical Assessment of Techniques for Protein Structure Prediction, shortly called CASP, which has been going on, I think, for 30 years now. So it's basically, it happens every 2 years, where the experts choose the variety of targets that then participants are predicting. So there is a variety in difficulty as well. And for the protein monomer prediction, the difficulty is already well defined, and it's based on the protein sequence similarity to the closest structure in the PDB. So before CASP 14, when AlphaFold 2 came up, there was this noticeable trend of not being able to predict difficult targets. And AlphaFold 2 was a breakthrough because it was, the first one that could actually predict also difficult targets and not just the easy ones. So the trend clearly changed. And that's important, you will see for my later point in the talk. And so deep learning is used for the protein structure prediction. And recently, mainly in the last year, there were quite some novel methods coming up for using deep learning for protein-ligand complex prediction. And those are so-called cofolding methods, because they simultaneously predict protein structure and the ligand pose from the protein sequence and the ligand smiles. So not as docking tools, which use the protein structure as the input. Here, you only need the protein sequence and to model both of them simultaneously. And there has been some benchmarking efforts for how to assess the performance of this method for the protein ligand complex prediction. Namely, there has been this postbusters set. There has been some efforts in the CASP 15 and also in the CASP 16. So here you can see the results reported in the AlphaFold 3 paper, in the CHI paper, and also in the CASP 16 conference. But there is one key missing aspect, which is how difficult actually is it to predict complexes in those data sets. So, and if with those data sets, we can actually detect a true breakthrough in the protein ligand complex prediction. And the answer is kind of no. And that's why we went also with this benchmarking data set. But the reason behind it is also because the target difficulty for protein ligand complex prediction is quite challenging to define. So there is no clear definition now. And basically, we believe that this definition should be based on the task. So the task of cofolding methods is to predict how the molecule A interacts with the molecule B. So the difficulty should be based on the similarity of interaction rather than what is now usually done. It's the similarity to molecule A. So if a protein is close to the protein sequence, and molecule B if a ligand is close by the Tanimoto similarity. So here are some examples why we think that the similarity of interaction is important to define the difficulty. So here in the middle one, you can see that you can have basically the same protein, but a different ligand that will bind in a different pocket. So basically, that is a difficult prediction because there is a new binding mode. Also, you can have the same ligand, but that binds to a different protein and in a different pocket. So that's again a difficult prediction. So that's why, so for all of these reasons, because yeah, until now we weren't really sure how to interpret the results of the performance on the post-buster set, on the cusp 15, the cusp 16. We decided that we need a benchmark that will truly assess the breakthroughs in the field, and that's why we introduce runs and poses. So what we needed to do in order to make this happen, it's basically we needed to collect and curate the protein-ligand complex data after the cofolding methods training cutoff, because everything before that is basically similar, so we cannot use that. And we need a reliable ground truth. As you will see that we also did some quality filtering. Then we also need to define finally this difficulty, novelty, similarity for the target complexes. And also we needed to define a success rate for predictions with cofolding methods, because it's not only now the ligand, but it's also a protein structure there. So yeah, this is our pipeline for dataset curation. So we took the complexes after the training cutoff of these methods, which is the 30th of September 21. We only took the X-ray structures and which feed some of the quality criteria. So they don't have any alternative configuration, and they are really a complex, and it's not just because of the crystal contacts. We also filter for the relevance, so there should be a certain amount of interactions that happen between the protein and the ligand. And at the end, we also did some redundancy filtering, so clustering based on the 80% of sequence identity and ligand identity. These are the numbers that we got. Finally, we got with 2,600 systems. And yeah, I will show the results based on the common subset. So not all of the methods produce the same amount of predictions, but we then compare just for the common subset of the modeled complexes. Then yeah, like I've been going on about the difficulty. So that was we needed to define that. So like I said before, difficulty should be assessed based on the similarity of interactions to the closest complex in the training set. So basically we have two main terms here. So from the protein side, we capture that with the binding pocket coverage. So basically, if your protein has a similar binding pocket that the target protein, and for the ligand similarity, we use this sucrose score, which basically measures the overlap of the ligand shape and the chemical features for the poses. I will show some examples. And yeah, so at the end, our similarity score is basically it's a multiplication between the pocket coverage and this sucrose score for the ligand. So we have the protein term and the ligand term. So this is how our similarity scoring compares to the, for example, protein sequence identity. Sorry for the small font, but so it's here in green. So for example, you can have something that by the protein sequence identity is quite far from the training set, but basically has the same binding pocket as seen here in dark blue. Maybe it will be better to show here on example. On the left, you have this example of two proteins. The target is this PDB, and the closest target was with this PDB. So these two complexes on the protein side, they only had sequence identity of 26%, but basically their pocket was the same, and also the ligand, as you can see, is almost the same and binds in the same fashion. So in our case, we can catch these edge cases and prevent the data leakage. And on the other hand, you can also have the low Tanimoto similarity and high similarity score. So to prevent the data leakage from the ligand point of view. So here you can see these two ligands that basically have a low Tanimoto score of 0.25, but they bind to the same pocket and in the same fashion again. So we can catch that with the Suko score, and the similarity then it's quite high because the pocket was exactly the same. And we also need to define a success rate for the protein-ligand complex prediction. So usually in the docking benchmarks, the one that it's used is the RMSD of less than 2 angstroms. But here, since we are assessing the cofolding methods, we don't care only about the ligand pose, but we also care about how the protein is modeled, and basically how the interactions with the ligand are modeled. So that's why we have also this LDT PLI term, which measures how well the interactions between the ligand atom and the protein residue are conserved. The RMSD, we have the binding pocket superposition symmetry corrected RMSD. So basically, you superpose based on the binding pocket residues, not on the global superposition. We saw that basically those two scores are complementary to each other. So that's why we are using both as a definition of a success rate. So basically, LDT PLI needs to be higher than 0.8, and RMSD needs to be lower than 2 angstroms for prediction to be a success. So we saw that sometimes you can have a good RMSD, quite low one, and a bad LDT PLI. So this is in the case on the left. Basically, as you can see, the prediction here, it's in white, it's in blue, sorry, and the ground truth is in white. You can see that by the global superposition, the ligand is basically, RMSD 0, but we can see that some of the binding pocket residues were not modeled correctly. So basically this loop is completely different in the prediction than in the ground truth, and we can catch that with the LDT PLI. And on the other hand, you can have low RMSD and good LDT PLI. This happens if most of the contacts are conserved still, but there are some binding pocket residues, as seen here in sticks, So white one again is the ground truth, and the blue one is the prediction. And that's when then after the superposition on the binding pocket, these two residues basically make the RMSD quite low. High, sorry. Yeah, quite bad. So yeah, finally the results. So after we have defined the success rate like we did, and after we have the similarity score and all of the complexes that fall into each bins, then we can run all of these deep learning cofolding methods on our test set. So on all of the 2600 systems. So we ran 4 of them, AlphaFold 3, ProteinX, Chai, and Bolt. And what we see, I mean, what's the key result here? It's that the methods struggle to do well on difficult cases. So the one that are the furthest away from their training data. And just a reminder, that was, what I was talking at the beginning. Basically, it is the same trend as was seen in the protein monomer structure prediction, where methods couldn't do well on the difficult cases, but could do well on easy ones. And AlphaFold 2 was a breakthrough just because it could do well on the difficult cases. So yeah, unfortunately, at the moment, none of the cofolding methods is actually a breakthrough. And yeah, then we also did some certification later on. Basically, we saw that in our common subset, we still have quite a lot of cofactors, for example, that can be found in many different pockets. And when we filter those out, the trend becomes even more linear. So yeah, the methods perform worse on the ligands that they see for the first time or just a few times.
Peter,
and also too,
we have a question on Q&A.
Hi. Yeah.
I think you should be able to see it. Yeah.
Yeah. Your benchmark. Yeah, we also considered Rosetta Fold all atom. I actually have it in the supplementary. The results were not great. Yeah, maybe I can show it quickly. The fold, yeah, it's here. So it was, it's quite low, I must say. So it's, yeah, I don't know if they've been working on it still, but yeah, it's not comparable to all of the AF3 related methods. So basically, it doesn't perform that well. We will update also the preprint with these results, but we didn't kind of want to take away the take-home message that is that still also the AF3 is not performing well on the difficult cases. So because then people usually just focus on the worst performing one. Let me go back. So yes, is there any other questions maybe? Not at the moment.
Nope, I think we're good.
Okay.
I see one more actually. I'm just gonna bring Smith on. Smith, feel free to unmute and ask your question.
Thank you. In your the slide 15, you showed that 2 cases of the good RMSD, bad LDDT, and the vice versa. So what is the global picture of your entire data set of the RMSD versus LDDT PLI?
Wait, I can. We have it in the preprint actually. Can you see my screen? Can you see the?
Yeah.
Yeah, perfect. It's in the supplementary. Wait. Yeah, it's here. So maybe I can. So it's so this is the basically the scores for the whole data set of RMSD versus the LDT PLI. So we can see that sometimes LDT PLI catches some of the cases where AlphaFold where RMSD is lower than 2. So yeah, and we inspect some of those cases, and usually it is that either there are added model contacts. So we penalize if there is a residue near the ligand in the prediction, which is not in the ground truth. So basically, those are called as added model contacts, which are also penalized. Because we have quite a reliable ground truth, so that's why we decided to go with that.
Okay, nice. Thank you.
Thank you.
Cool. I think we have one more question from Cesar.
Yeah.
Yeah. Can you hear me?
Yes.
Yep.
Yeah, very interesting work. I mean, having also playing around, well, just playing really, not doing a benchmarking, but I have noticed what you have seen here on Rosetta Fold all atoms. It works really bad on most of the predictions. But I was wondering if you have checked, I think there is a paper published recently by Exentia somewhere that they also checked if the interactions are recovered from the predictions. That would be another test that might be interesting to see.
We are also working on that so it's with the PLIF, right?
Yeah, I think so. Yeah.
We will. It's an ongoing stuff. So yeah.
Because I guess it's gonna be even more difficult to recover all those interactions.
Yeah.
Because in my case, I have seen I need to work a bit more. If I do a prediction, I need to work a bit more, maybe doing a relaxation outside in another software to make it better, because I can see interactions are not well recovered in most of them.
Yes. So we also have maybe I have it here even. The plot just with the if we define a success rate just with RMSD being less than 2, so we see that the success rate gets a bit higher, but the trend is still there.
Okay, in this plot, when you say, I'm a bit lost when you say similarity to a training set, it means in terms of the protein or the ligand?
It's basically both in terms of the protein based on the binding pocket, and also in terms of the ligand based on its shape and chemical features.
How do you combine to make these bins?
So we multiply those two terms. I have it here.
Right. Yeah.
Yes. So basically, the first part here, it's based on the protein. So we did it with the fold-seq superposition, and if the binding pocket is aligned to the binding pocket of the target, so for the residue, so we get kind of the coverage of the binding pocket. And the sucrose basically this part is the measure for volume overlap, and the score fit. So it's the RDKit functions, and the score fit is measured for the here it's written wrongly. It's measured for the chemical features overlap.
Mhm. Okay, perfect. Thank you.
Where did I was here, yeah, I think. Or are there any other questions maybe at this point?
No, nothing else in the chat, so feel free to keep going. Thanks.
Okay. So yeah, so like I said, so we did the cofactor filtering. So we saw that the performance of these methods is better if they saw a ligand multiple times in different binding pockets. So we can say that this methods works the best for cofactor-like ligands. But while, yeah, for the drug-like, we see the trend being more linear. But just to make sure that we also account for the over-representation of certain protein families, we also analyzed only the cluster representative of drug-like systems. So basically here you can see that the numbers are more evenly spread than in these cases, because here, yeah, there are much more of highly similar complexes that are of dissimilar ones. And yeah, there are some selected examples that I want to show that you also can get a feel for the similarity score. So here on the top left, it's an example from the lowest bin. So where the similarity is less than 20, we can see that the ground truth and the closest training system are, so they have the same protein, but the ligand is bound in a totally different pocket. So the pocket coverage is quite low, and also the succose is quite low. So it comes down to low similarity, and also, yeah, we saw that prediction of all of the methods were wrong in this case. Here it's the one from the 20 to 40 bin. It's a kinase where basically the ligand binds into this DFG out conformation of a ligand, but the predicted conformation was DFG in, and in a different pocket as well. So yeah, the scores were quite low for that one as well. What was interesting to see, it's also that sometimes these methods don't really care about the chirality that you put your input smiles in. So basically, our input smiles had the information about the chirality, but all of the predictions kind of orient this chloro group into different direction, try to mimic the closest training system, and of course this prediction was also a failure for all of the methods. And yeah, there were some cases also of failures in the highest bin. Yeah. So we also ask if, you know, if it's a problem maybe of if it's a problem of not finding the right pose, or just not ranking it correctly, because we only compared in the previous graph the top ranked prediction from the methods. So basically we then scored all of the 25 models predicted by each method for each system, and we saw that the ranking is not perfect. So you can see the difference between the top and the best, so the black and the blue one. So it can produce better poses, but it's not only that. So sometimes it doesn't sample the best pose even. What was also interesting to see is that the multiple seeds didn't have an impact here for this task. So you can see that the black, which is top out of 25, and the gray top out of 5 are basically overlapped. So sometimes you don't even see the gray line. So here we could only go with producing 5 models with 1 seed, and the results would be quite similar. And yes, there are some ongoing activities. So what we want to do is to compare also these cofolding methods with the physics-based methods. So there are different scenarios that one can use the docking methods. So one that it's quite idealized is the redocking. So you have perfect conformation of a protein, you know where the binding pocket is, so that's that's the redocking score, and we saw that there is no obvious trend of the docking methods, which is a good thing, but we also want to try a more realistic scenario where we would be docking a ligand into the closest template and not the ground truth structure. So is there hope? So yeah, we developed this benchmark in order to detect a breakthrough in the protein-ligand complex prediction. So for now, this breakthrough didn't happen yet with the current generation of cofolding methods, because we saw that they struggle to generalize to unseen data. And yeah, there are some questions of what can be done to change that. Basically, algorithms seem not to be really fit for the purpose. So they don't include any of the physics terms yet. So what are the models actually learning? Because if you consider how AlphaFold 2 worked, is like it learned the context from the MSA, and in the so for the ligands we don't have that co-evolutionary signal, and we don't know yet how to find the signal in the data. But yeah, what can be also done, it's some data augmentation tactics, so trying to fit it with the ligand density instead of just the coordinates itself. So there are different ways that methods can evolve. And so like I said, we developed this runs and poses benchmark to find the breakthrough for the protein-ligand complex prediction, and it's meant for benchmarking the methods with the training cutoff of 30th of September of 2021, because that's how the similarity scores are computed based on that date. And also thanks to Polaris, where this data set and benchmark are ML ready, so you can test your method for protein ligand prediction just by installing their Python library, and you don't need to care about the parsing of the stuff. You can also get all of the data in our Zenodo website, and you can also check our GitHub repo to see how did we plot the graphs and everything, and how did we compute the similarity scores. The models will be also soon available in the model archive database. And here it's again the QR codes to the preprint GitHub repo and Polaris benchmark. And yeah, with that, I would like to thank, first of all, my co-authors, so Jerome, Torsten, and Jay. Yeah, was a pleasure to work with you guys. And all of the Schwede group, and especially to the subgroup of Picky Binders, and all of our collaborators. And yeah, thanks again to Johnny for making this webinar happen.
Cool, awesome. Thanks, Peter. Yeah, runs and poses, Picky Blinders. You guys have really, I think, maybe one of the best naming conventions in the
With the
space.
names, we can be quite creative. Yeah, that's true.
Cool. Okay, I think we have a few questions actually in the chat here, just in the Q&A part. So I think the first question was, I've tried bolts one and it seems to change double bonds to single bonds in some of the cases. Is this kind of modification something you've noticed on a bigger scale besides chirality?
We mostly notice chirality issue. So the problem could be also that so I don't know if bolts outputs the bond connectivity in their C files. I know that AlphaFold 3 doesn't, but we didn't notice that. We haven't filtered allosteric binders. So for now, yeah, I think that all of these complexes are still quite useful sometimes. You know, sometimes in the allosteric places is also where magic happens. So no, we didn't do any filtering in by the allosteric binding pockets. Could you clarify the similarity on the figure one as it? Okay, let's go to figure one. The question was, yes. Yeah, so basically how did we find the cofactors to filter for is, so we looked at only the ligand, and we compared by the Tanimoto similarity, so basically by the ID, to anything in the training set. So if it occurred more than 100 times, so for example, like ATP, you will find, yeah, for sure more than 100 times in different binding pocket. So with that filtering, we catch those cofactors. If that answered the question, hopefully. Docking, we used Smina and Nina. Yes, those two. A general question. That may. Yeah, so that's the question about the potency usually comes up. So can we actually, based on the protein-ligand complex prediction, infer also potency? The answer to that one is no now. So basically during CASP16 there was an experiment where participants needed to predict a complex, but then also to rank it by the potency and to rank it based on their prediction, and later then in the stage 2 also based on the ground truth. And what was quite sad is that also when given the ground truth, we cannot rank the ligands when we know how the structure looks like. We cannot rank them by the potency. So that was a bit sad.
Cool. I guess I'll take the next question. Yes, we're recording this, and we'll be putting it available on the Polaris YouTube, and we'll also post it on our socials. So you'll probably see us release this early next week. Okay, I think there's one question from Cesar as well. I'm just gonna, Cesar, if you want to go off mute, you can ask your question.
Hi, thank you. So yeah, I mean, this is very interesting, but I was wondering if you have some thoughts about if we will be able to reach some generalization from these models. Is there any signal that show us something like that, or just they are relying on the training data set, most of the prediction that perform well?
Yeah, so for now it seems like they mostly rely on the similarity to the training data. So I believe what it needs to happen is that we need to change the architecture or the algorithm a bit to be able to actually catch what is going on, not to just memorize the pose. But yeah, there is I don't know if I'm the person to answer this, but yeah, there are some strategies that all of these models can be improved, and I think they will sooner or later. So yeah, I think there is hope for it to generalize.
Because at the end, when we have, let's say we have a complex we want to predict, and then we can just look for what is similar in the data sets and perhaps make an emerging model, something we have been doing for a long time, not running.
And that's why we also want to make these baselines, basically, because right now the way to go is to do the homology modeling and or the template docking. So this year, actually on CASP16, the group that performed the best was basically on the template docking. So basically they checked what is similar in the PDB, and then they used different bits of the ligand to put it together. So yeah, so that's why we also still want to do the baseline to kind of see if there was any improvement actually.
But I remember Smina and Nina are scoring function methods or are AI methods. I don't remember.
No, they are docking methods. Nina, I think the scoring is then done with the deep learning, but not finding the pose, just just scoring of the pose. And Smina, there is no AI.
Okay, thank you. Nice talk.
No, thanks.
Okay, I think we have another question just in the chat. I'm just going to read it out so that it's easier for people that watch the recording. They can have a better, I guess, understanding of the question. But yeah, so are you going to update your model to include structures solved since September 2021? And what is the diversity of structures in the model set? For example, you mentioned kinases. Does the model do better with kinases because they are represented more in the model training set?
So if I understand the question correctly, So basically we have so our set is after September 21. It just, if you want to assess the, the difficulty or the similarity to the training set, we can compare it to that date when the methods were training on. And the diversity of structure. So yeah, so we did the clustering of the structures also. I think we get, I don't know, something around 700 clusters or maybe less. I will need to check that again. But yeah, the two highest clusters were the SARS-CoV MPRO structure, because there was quite a lot of efforts done to solve a lot of structures there, like moonshot. So that was the biggest cluster, and the second biggest was the kinase cluster. So yeah, there's a lot of data for kinases, but the issue is that if there is a kinase with a novel binder, the methods don't perform well on those. So that's the issue we found, because yeah, like in the drug the pharmaceutical companies, they would usually work on kinases, but with a different type of binders. And even for novel binders, it couldn't find the right pose. So that was a bit worrying. But most of the kinases anyway fall into the highly similar, because there are some of the ligands that have been solved in many of kinase structures. The next question is if we try to dock to apo structure instead of redocking. Yeah, so that's what I have written here in the realistic scenario. So realistically, you wouldn't have the ground truth structure, so you wouldn't be able to do redocking. But what you would usually do is you would check in the PDB what's the closest template that you have. Either with that ligand, maybe with a different with the protein that's in the same family, but it's different, and you would use that protein to model your protein, then dock it. So that's what we are doing at the moment to have more of a fair comparison. Yeah. Thank you.
All right. Any other questions? Feel free to raise your hand and we can bring you off mute. Okay. If there's no more questions, I just wanted to share quickly that Polaris right now, we're actually running a competition in collaboration with the folks at ASAP Discovery and OMSF. And so, as part of the competition, there are 3 sub-challenges. One of them is on predicting ligand poses. So if this is something that you're interested in, the competition is actually running right now and is still open for another two weeks or so. You'll actually be working with new sort of training data released from a program that ASAP has been running actually on the MPRO target. So there's more details here in the link that I'm going to share just in the chat. So if you're interested in participating in that, the other 2 sub-challenges are on potency as well as admit property prediction. So there's more details in the link there if you're interested in participating. I know Yankee is here in the chat who is part of ASAP. So Yankee, feel free to chime in if there's any other thoughts you want to share about the competition right now.
Yeah, thanks, Johnny. I don't really have much to add. I think the main thing that I wanted to say is that we're really hoping that more people will participate in the posing challenge, because that really was the most difficult thing for us in the actual drug discovery process as well. So a lot of the predicting the admet and the potency was fairly straightforward because we had the internal data, but the posing specifically was really difficult because we had a lack of X-ray data for quite a while. And we're interestingly also kind of seeing some parallels with what Peter is presenting, where we actually created that ideal situation with redocking with a closest template, and that seems to work much better than a cofolding model. So we'd really like to see participants actually try to outperform our base models on that.
Cool. Awesome. Well, thank you so much, Peter, for taking the time to do the webinar and share more about your work. I think it's really awesome, really exciting. And thank you to everyone who tuned into the webinar. We'll be releasing the recording as well for folks to watch afterwards. So thanks, everyone. Have a great weekend and see you around.
Thank you. See you.
Thanks.